Chapter 15. And Chapter 15, we're going to be talking about microbial mechanisms of pathogenicity. So we're going to be actually talking about micro a little bit. So for a famous pathological microbiologist there, Rebecca Lansfield, made lots of contributions to that world. So here we are. So we're going to be discussing a variety of things here about bacteria, virulence, a little bit about virulence factors. We won't get too far into virulence factors. We won't have to learn the different virulence factors for different bacteria. It's not like we're going to be learning about yachts and all this stuff, but we will be talking about a variety of things, and there are some virulence factors at least discussed here. Basically, it's about portals of entry, how we get things in. Microbes enter the body through mucous membranes. They can enter like the respiratory tract, the GI tract, the genitourinary tract, or the conjunctiva. So they can enter mucous membranes of our body. So basically what we do is we touch something, a surface that somebody got a bacteria on or a virus on. We then touch our face, our eyes. We breathe it in, something like that. Integument skin through hair follicles, sweat gland ducts. They actually, some things bore through the skin, including some insects can bore through the skin and then insert microbes. The parenteral route under the skin, like an injection, like a bite, insect bite, a puncture, an injection, a surgery wound, things like that. Preferred portal of entry is just the occurrence depends on the of disease depends on if the occurrence of disease depends on the portal of entry. So sometimes you won't get a disease if you don't get it in the right place. You know, a great example of this is when we have things like stepping on a nail for tetanus. Okay, all right. Now, I want to talk about the infectious dose and the lethal dose. What we call the infectious dose, the ID50. Infectious dose, 50% of a sample population, describes the virulence. And the lethal dose is lethal dose for 50% of a sample population. That describes potency, like of a toxin or something. How bad is it? There are ways, bacteria to make us sick, they need to adhere to another person's plasma membrane. So attachment of a microbe or phagocyte to another plasma membrane is called adherence. So they can form biofilms, allow for pathogenicity. They have to stick to make us sick. Okay. There's things called adhesins. These are ligands or carbohydrate-specific binding proteins that stick out of prokaryotic cells. For example, in Streptococcus mutans, they'll use the glycocalyx. Escherichia coli will use a fimbriae. Streptococcus pyogenes will use an M protein. And then there's, on the host cell that binds to an adhesin is called a receptor. So basically we have our adhesin molecule on our pathogen and our receptor, and these things can stick. And if they can stick, one of the things that antibodies can do is coat this bacteria and prevent this sticking from happening. So if it can't stick to its target receptor and bind to us and adhere, it can't make us sick. And there's a variety of ways it can do that with different secretion systems and being able to release toxins, be able to release things inside, things like that. So these adhesins are really important. All right, now they can penetrate our host defenses with capsules, prevent phagocytosis, so they can get past our defenses, our phagocytes, like Streptococcus pneumoniae, Haemophilus influenza, and the Bacillus anthracus, all encapsulated bacteria that do that. Cell wall components like M protein found on Strep pyogenes can resist phagocytosis. There's OPA proteins, they inhibit helper T cells, like found on Neisseria gonorrhea. That's what causes gonorrhea, Neisseria gonorrhea. Mycolic acids, waxy lipids, resist digestion on Mycobacterium tuberculosis that causes tuberculosis. There's enzymes. Virulence of bacteria can be aided by exoenzymes like coagulases that actually clot fibrinogen in blood. They cause clots. They help isolate and protect us from the host defenses. There's an IgA protease. IgAs attack before you enter. They're going to be found in things like saliva and tears. So they break down the IgAs, antibody and secretions like saliva, tears, things like that. Enzymes that also help microorganisms spread from their site. There's kinases to break down fibrin and digest clots to help them escape. There's hyaluronidases. They hydrolyze hyaluronic acid in connective tissues. And there's collagenases. They break down collagen in connective tissues, and this lets it spread. Okay. There can be antigenic variation, changes in the surface antigens that occur in a microbial population that allows it to become unaffected by antibodies. The antigens that the antibodies are now made against, so a lot of you at least have covered this in AMP2 by now, or if you're in AMP2 while you're taking this, you've learned this in AMP2, is you make antibodies against antigens. If we can vary the antigens on surfaces of things and make new proteins on the plasma membranes with alternate genes that we have in bacteria or let's say a protozoan, we can now evade the antibodies that we're making. Influenza virus does this. Neisseria gonorrhea, Trypanosoma brucei, the subspecies Gambinesi, that causes African sleeping sickness. So, for example, after infection, you will start to see new clones showing up with new antigenic variations. Okay, new clones. Other ways to penetrate the host cells, get through the cytoskeleton. We can use invasins. There's surface proteins that actually rearrange the nearby actin filaments in the cytoskeleton. So Salmonella, Typhimurium, and E. coli can do this. Membrane ruffling to disrupt the cytoskeleton of the host cell. Microbes can sink into the ruffle and be engulfed by the whole cell and get in through membrane ruffling. Actin can use to get to actually propel microbes through the cytoplasm to jump from one cell to another. Listeria and Shigella do this. So this example, basically ruffling, getting in. Okay, that's actually showing it happen. Now, bacteria can damage host cells. They can actually use the host nutrients. So one of the things we can do is you have valuable nutrients like iron or other nutrients, and we can take those nutrients away from the host cell and use it ourselves. We can cause direct damage in the immediate vicinity of the invasion. There's things like toxins that can be that things damage things through like type 2 secretion systems that can be secreted, type 3 secretion systems. We can produce toxins and transport by blood and lymph that damage sites far from the invasion. So toxins can go out and you step on it, next thing you know it's affecting your jaw muscles. Okay, introducing what's called inducing hypersensitive reactions, the hypersensitivities. I talk about type 1, type 2, type 3, type 4. Talk about the hypersensitivities that we get in AMP2 a little bit. Most bacteria do need iron. Why do they need iron? They need it for cellular respiration. In the electron transport chain, most of the proteins in the electron transport chain have iron in it. They're called cytochromes, and they have iron in it. And so we need siderophores. Free iron in the body is mostly bound to carriers, like for us, transferrin. In bacterial cells, we use siderophores, bacteria iron binding proteins. They secrete it, they take iron from the carrier, it goes in the bacteria, it's taken up, not always, sometimes toxins to kill the host cell, and then you just release iron. So we're stealing iron from cells. Bacteria like to steal iron, they need it, so they're pumping iron with siderophores. I had a friend of mine who had a t-shirt with a siderophore structure on it, and it said pumping iron with siderophores. And it says Anyway, siderophores are iron carriers, iron pumpers. Okay, we can do direct damage. We can grow outside the host cell. We can use nutrients. We can we can rupture the cell. We can spread to other tissues in greater numbers. Others get uptake through phagocytosis, then disrupt the host cell. They basically get released through a reverse of phagocytosis, and they can enter other cells. But most damage is done by toxins. So most of this damage is toxins, as I mentioned. Toxins are poisonous substances made by certain microbes. They can induce a lot of things like fever, cardiovascular issue, severe diarrhea, shock. They can destroy protein synthesis, destroy red blood cells, cause hemolysis, blood vessel damage, disrupt nervous system damage, causing spasms, neurological damage, things like that. There's about 220 no bacterial toxins since I wrote this. And 40% of disease cause disease by damaging the eukaryotic cell membrane. So they directly damage it. Toxigenicity is the capacity of a microbe to produce toxin. Toxemia is having toxins in your blood. Okay. These toxins can be exotoxins, and these are proteins that are produced inside the bacteria. It's part of their growth and metabolism. It gets secreted in the medium through one of the secretion systems of a bacteria, diffused into blood, gets transported through the body, and destroys host cells somewhere else to inhibit metabolic functions. It may make the cell not do certain things. We'll talk about exotoxins. Usually, exotoxins is what the disease is caused by, not the bacteria, but the toxin the bacteria releases. Antitoxins can be antibodies produced by the body to actually destroy and provide immunity from the exotoxins. Toxoid is an inactive toxin. We can so we do toxoid vaccinations, a toxoid vaccine, like with diphtheria and tetanus. We don't actually go against the bacteria, we go against the toxin that diphtheria produces and tetanus produces. Okay, the diphtheria toxin and the tetanus toxin, the bacteria that make diphtheria and tetanus happen. Okay, and we produce immunity with the toxins. The exotoxins are named for the type of host cell that's attacked. For example, a neurotoxin if it's neural tissue, cardiotoxin if it's heart tissue, hepatotoxin if it's liver tissues, leukotoxin if you're dealing with lymphatic lymphoid tissues, and terotoxins got cytotoxin cells. For example, the diphtheria toxin and the tetanus toxin for the disease that it causes, or named after a specific bacterium like botulinum toxin from Botox, the Botox toxin, botulinum toxin from the botulinum bacteria, Vibrio enterotoxin like Vibrio cholera, makes you poop your guts out. There's AB toxins. These are two-part. The A part's an active enzyme, and the B part is binding. The B part binds and allows the A part to enter, and the A part's an active enzyme that actually disrupts the metabolism. There's membrane disrupting toxins. They lyse the host cells to create protein channels in the plasma membrane and allow the bacteria to escape. This allows it to spread. There's superantigens. Superantigens cause intense immune response. Host cells can release cytokines, and that stimulates proliferation of T cells. And then we start getting fever, nausea, vomiting, diarrhea, shock, and even death because of all these cytokines. Okay, cytokine storms and things like that. So AB toxin, what happens is our host cell, it makes an AB toxin. The A part is the active, the B part's the binding. The B part binds, the A part enters. The A part enters and starts to damage the inside, altering the function, and protein synthesis or enzymatic activity does something to damage that cell, and now the cell can't make crucial proteins. So, for example, exotoxins, we have botulism, Clostridium botulinum. There's an AB endotoxin. It's a neurotoxin, actually prevents the transmission of nerve impulses, causing flaccid paralysis. So if your neuromuscular junction cannot transmit to the muscle, you're flaccid paralysis. Clostridium tetani is an AB toxin it causes tetanus. Neurotoxin blocks nerve impulses to muscle, causes relaxation, uncontrollable muscle contractions. Okay. Diphtheria, Corynebacterium diphtheriae, AB toxin. It's a cytotoxin, inhibits protein synthesis in the nerve, heart, and kidneys, causing diphtheria. Scalded skin, which happens in Staphylococcus aureus, is an AB toxin. It causes the skin to slough off, called scalded skin. It damages the cell adhesion molecules. Cholera, Vibrio cholerae, AB toxin. It's an enterotoxin that actually secretes large amounts of fluids and electrolytes, cause you to poop your guts out. Travels diarrhea is the usually caused by E. coli gastroenteritis. Could be Shigella. It's an AB toxin. It's a enterotoxin. Large amounts of fluids and electrolytes, diarrhea, you poop your guts out. Anthrax, Bacillus anthracus, AB toxin. The two A components enter the cell and via the same B, and the A proteins cause shock and reduce immune response. So some can cause really bad immunosuppression. We have membrane-disrupting gas gangrene and food poisoning, or membrane-disrupting toxins. So Clostridium perfringens and other species of Clostridium cause gas gangrene and food poisoning. There's a cytotoxin, massive red blood cell destruction, hemolysis. The enterotoxin will cause food poisoning, cause diarrhea, poop your guts out. The antibiotic-associated diarrhea, Clostridium C. diff, Clostridium difficile. Membrane-disrupting toxin, enterotoxin causes you to secrete fluids, electrolytes, to severe diarrhea, and a cytotoxin disrupts the host cytoskeleton, causing damage. Food poisoning can be caused commonly by Staphylococcus aureus, which is a super antigen, secretion of fluids and electrolytes causing diarrhea, and toxic shock syndrome by Staph aureus, a super antigen. Again, fluids and electrolytes, and damaging results in capillaries. Blood volume, lowers blood pressure, causing toxic shock. This can be happen if you leave a tampon in for too long and get toxic shock. Okay. I like to talk about these. I currently don't have those on the quiz, but I always like to talk about them and go over them to give you guys an idea of the importance of why these AB toxins, membrane disrupting toxins, and super antigens matter so much. Okay. Endotoxins, there's a lipid portion, lipid A, of a lipid polysaccharide, LPS. It's part of the outer membrane of gram-negative bacteria. When the bacteria die or the cell wall undergoes lysis, it's released. They produce signs and symptoms of chills, fever, weakness, aches, sometimes even shock and even death and even a miscarriage. Although not to the same degree. But the thing is, this can set off complement cascade and set off other parts of our non-specific defenses. Endotoxins can be very pathogenic. It can be disseminated through intravascular coagulation, what we call DIC, activation of blood clotting proteins, small blood clots, slower blood supply to tissues. Endotoxic shock, this is life-threatening, decrease in blood pressure, capillary permeability happens, our fluid leaves our blood vessels. This is what Salmonella typhi does, really bad. This is what Proteus species do, and Neisseria meningitis does. So what can happen is endotoxins and the pyrogenic response is we get the bacteria has an endotoxin, we release cytokines. These cytokines act as a pyrogen, go to the hypothalamus sees these prostaglandins, these pyrogens, and they are going to go to the thermostat of the brain, the hypothalamus, and tell us we need to raise the body temperature and get a fever that can be very bad. Fungi can also have pathological effects. Fungal waste products can cause symptoms. They can be chronic infections to give allergic responses. For example, there's toxins that inhibit protein synthesis, trichothecene toxins like from fusarium. There's proteases from Candida and Trichophyton, and there's capsule can prevent phagocytosis in Cryptococcus. One that I want to talk about is Claviceps purpura. That's a fungus that grows in grains like barley. It produces ergot, which is a toxin. Ergot, called ergotism in the Middle Ages. People used to blame this for what they think was causing the witch trials throughout, is that ergotism caused hallucinations, and people thought they saw Temperance Prudence dancing with the devil in the pale moonlight. They were hallucinating. They were high on acid. This is where we get LSD. It causes hallucinations, delirium, and psychosis. It grew on rye, for example, cereal grains. People made bread out of it. They eat it, and they got a little high. It's a natural source for LSD. Modern milling usually prevents this, but people back in the old days would get it. Aspergillus flavus is a mold that sometimes grows in peanut butter. Produces what's called an aflatoxin. It's carcinogenic. It can alter the body, produce mutations. Amanita phalloides, the death cap, produces phalloidin and amantine. Okay, that's a neurotoxin that affects our autonomic nervous system. If we ingest it, can kill you. One that's really badly neurotoxic is also found in Amanita muscaria. That makes you feel like you're getting big, and we think that's to blame for Santa and his reindeer. It's a red mushroom with white dots on it, and when reindeer eat it, they jump really high because they feel like they're bigger, and they feel like they have to jump over things. So it's Santa and his reindeer and the flying reindeer. You also apparently see little people when you're on it. So Santa Claus and his elves and his flying reindeer probably came from mushroom high. There's actually Aspergillus flavus and Amanita phalloides. Plasmodium can reproduce in the host's red blood cell, causing the lice like in malaria. Giardia lamblia attach to host cells like by sucking disc and digest cells and tissue fluids. This causes what we call beaver fever. Okay, so this is the Plasmodium living in red blood cell. And this is a Giardia lamblia trophozoite. Look how he's smiling at you. He looks really happy. Pathological effects of helminth and algae. Wookiera. There we go. Wookiera bancrofti is a roundworm. This is always a hard one for me to say, is Wookiera bancrofti. It's always been a hard one for me to say. I'm not particularly great with my helminths. Not my main area of expertise. I took invertebrate zoology and learned many of these species and what they did, but I always wanted to take a parasitology course. They block lymph circulation, so lymphocytes, we call that elephantiasis, grotesque swelling. I'm not going to show really nasty pictures of elephantiasis yet. I think I have some in the diseases section. Alexandrium is an algae that makes a neurotoxin called saxitoxin. They're fed on by mollusks. People eat the mollusks, and it causes paralytic shellfish poisoning. So you got to be careful with some of these. Some of these algae produce neurotoxins like the red tide and things like that. Portals of exit, basically we poop out a lot of things, is one way. We poop it out. Poop and pee it out is how a lot of things leave. We can cough and sneeze it. We can poop and salivate it out. We can urinate it and excrete it vaginally. We can, through our skin, it can be excreted under our skin. Through blood, a biting insect, or needles and syringes. We have a disease. We use needles. We get bit by a bug and they transmit it. Things like that. So pathogenicity, portals of entry. We have things that can invade or adhere. They penetrate and evade or get past our host defenses. They damage our cells. Then they exit and go off to affect the next person. Okay? Basically, that's how it works. We have portals of entry, mucous membranes, or respiratory tract, gastrointestinal tract, or genitourinary tract, our conjunctiva, our skin, and then if I go in and get stuck with a needle or something like that that's dirty. Microbes invade. How many of them, and can they adhere? Because they have capsules, cell wall components. They have enzymes, antigenic variations, and invasins and intracellular growth can allow to invade and adhere. Siderophores and direct damage, toxins, endotoxins and exotoxins. I didn't talk much about lysogenic conversion and cytopathic effects that damage the cell. There's also portal of exit, same portals given for the entry for many of them, depending on the microbe. Okay? Alright, that finishes it, and we learned a lot about how we get sick from microbes. Thank you, and see you in the next one.